Um, okay, so we are going to progress through the ADA 2007 free full-length exam. And just as a reminder, I'll just do this for a moment to remind you that these are indeed real retired questions. So these are questions that were operational on the real exam. Now, are any of these questions from 2007 relevant to the exam that you will be taking? Yes, apparently. So the ADA in their 2022 exam. So this obviously is an exam that you can have access to, or you will always be able to have access to the newest version because they're available through, it is available, not there. This singular 2022 exam is available through Prometric, and you can still get linked to it from the ADA's website. And it is composed of some precisely the same questions, except one answer choice removed, minus E. For many of the answer choices in the new DAT have much less of the 5 answer choices than it used to have. So it has far more of the 4 answer choices for the natural sciences, that is. And it is composed of questions from the ADA's 2007 exam, which we're doing, and the 2009 exam, which is no longer in print because there were some issues with it. However, if you do get access to it, it is much more challenging than the 2007 exam for most sections. And then a little magic formula. The ADA added some additional questions. And as we said in the past, there were new topics added to biology. And so adding new questions and also adding some more challenging questions creates the new 2022 exam. So yes, the 2007 exam is relatively easy, but that does not mean that none of the questions could be on the real exam. None of those, that level of difficulty could be on the real exam because the ADA has established that some of those questions are good enough even still now. Okay, and not to mention, it's a good exam to just start your warm up, getting used to seeing the depth and breadth of different questions and starting easier. So because it's relatively easy, I wouldn't say that 2007 exam should be used as a diagnostic or prognostic exam to see what kind of score you'll really get. No way. But it is a good introduction to your studying and being disciplined about progressing through the practice questions. So the natural sciences, of course, 100 questions, 90 minutes, which means 54 seconds per question. When you're taking the real exam, the standard approach is to spend 30 seconds per question for biology. So half a minute each. So there are 40 biology questions out of the 100 questions in natural sciences, the rest being equally split between general and organic chemistry. And spending half a minute because you can recognize the answer relatively quickly. And in general chemistry, you'll have to do calculations. And in organic chemistry, occasionally you'll be doing some structural drawings but there's more reasoning, more thinking in organic chemistry. And one more thing. Let's start. Okay, so you have the link to the full-length exam, and we already did in our last webinar questions 1 to 5. We're going to be doing 6 to 10. Again, relatively easy. But when you are looking at the questions, if you have not seen the questions before, then try to work through it, think through it, answer as best as you can. But if you find that you did not review that content before, for whatever reason, it's really basic, but for whatever reason, if you haven't done that before, then please Google, call a friend, call a professor, whatever, but do anything you want to come anything you can to come to a solution without looking at the answer first, because just thinking through content is always more valuable than just looking at a solution. And of course, aside from the fact that these questions are from the ADA, but the ADA also produces the questions not only for the DAT, but also for the OAT. And they're the identical questions for the OAT, just the marking scheme and the scale is different. Okay, so we're going to do questions 6 to 10. You will have 5 minutes, so 1 minute per question, because we're getting warmed up and all of that, and we're not at the stage that you're ready to take the real exam, when you'll be doing half a minute per question. So it'll be 1 minute per question. You'll have 5 minutes. Then we come back. Then we'll go through the solutions and try to give a little background information where relevant, so that you can answer more difficult questions and questions related to those topics, because yeah, that's an important point. The ADA chose these topics for a reason, and many of the topics recur over time, including usually the balance of the exam and types of questions on the exam as well. So that makes it helpful. Okay, so let me just get a timer here. So you will have 5 minutes to complete it, and then we will talk about it. Please begin, and good luck. Okay, so that is your 5 minutes. I hope it was not torture. I hope you found that they were pretty straightforward, but I'll try to add some information to that from time to time. Okay, so the first question. We have intercellular organelles that participate in metabolic oxidation involving H2O2. So, yeah. So this is hydrogen peroxide, which hopefully you are very familiar with because of organic chemistry. It is famously used in all kinds of reactions in organic chemistry to oxidize compounds, or even because it's hydrogen peroxide peroxides generally are very famous for having a oxygen single bond. They can engage in free radical reactions. So with light energy being provided, HF, light energy, it can create free radical reactions. But that's for a different day. That's when we're going to be talking about organic chemistry down the road. So anyway, but just by recognizing it's hydrogen peroxide and then looking at the answer choices and having the answer choice peroxisome, the only way you get this wrong is if you don't recognize hydrogen peroxide, or if you think it's a trap. And yeah, the ADA doesn't really usually have traps. I mean, it's more you know it or you don't know it. So these are a list of organelles, that is being presented. The first one being centrioles, which, of course, is an organelle, but it is not membrane bound. Perhaps we're going to be discussing that at some point. But it is an organelle, sort of a cylindrical, you can imagine it that way. It's mostly composed of tubulin, and they're usually in pairs. The centrioles are usually in pairs and are very important in terms of mitosis, meiosis, and the spindle fibers. And if you imagine that this cylinder, a very common way of remembering centrioles is that they have a certain 9 sets of microtubular triplets. So 9 sets of triplets. So that's usually how it goes. And then answer choice B is endoplasmic granules. Endoplasmic granules, what is that? So endo means inside. Plasmic refers to plasm, which is it's in the cytoplasm, but it is composed of a plasma membrane. It's surrounded by plasma membrane, and granules makes you think of something that's granular, of course, like sand or something like that. And in the cell, that which looks granular is the rough endoplasmic reticulum, because it's studded with all these ribosomes. So it appears granular on the light microscope, or even electron microscope. And so we are thinking in terms of some something that is associated with the rough endoplasmic reticulum. And then, of course, answer choice C, which we said is the correct answer. And then lysosomes, those are, some people like to remember lysosomes as suicide sacs because they contain the digestive enzymes for the cell. So they can combine, going from primary to secondary lysosome with some old cellular part or something that needs digestion, and then fuse, and then its enzymes can just tear it apart into some basic molecules that can be reused. So even if you're a cell is infected with a virus, a lysosome can fuse with some of the, if it can recognize the foreign nature of it, and digest it into its components, so that you can use the amino acids and other components, basic carbohydrates of the virus, in order to make better things for your cells. And then finally, macrobodies, which is not really a thing, but macrobodies, okay, so the ADA might have put a little trap in there for this version, because it might make people think of microbodies. And that is a term that can be applied to peroxisomes. So peroxisomes is part of a part of the organelles that can be referred to as microbody, but not macrobodies, which is just like a play on words. But okay, back to peroxisomes. So what does it do? Well, obviously, hydrogen peroxide is famous for oxidizing things in organic chemistry. So we would expect that peroxisomes are involved in oxidation reactions. And indeed they are. They're involved in oxidative deamination of amino acids, oxidation of long chain fatty acids, and also cholesterol synthesis. But I want to just underline this oxidation thing again, because it did come up last week. Just some ideas to keep in mind. Remember that from general chemistry point of view, loss, electrons, oxidation, gain, electrons, reduction, that's general chemistry. We are more in the biochemistry world. There's tiny amount of that on for the DAT. And so what we are thinking in terms of is that oxidation is a gain of oxygen content for a molecule, or the loss of hydrogen content, and reduction is the opposite. So this is something you can keep in mind for both biology, obviously, because of the subcomponent of biochemistry, and for organic chemistry, and this comes up even more often in organic chemistry. Reduction is the loss of oxygen content. Perfectly logical. Oxidation is the gain of oxygen content. Reduction is the loss of oxygen content, or the gain of hydrogen content. So what just happened? In the peroxisome with hydrogen peroxide, there is oxidative deamination, there's oxidation of long chain fatty acids. So then if you oxidize something, you are yourself reduced. This is a famous little twist that sometimes happens in general chemistry. The oxidizing agent is itself reduced. Okay, because these things happen together. That's why it's called a redox reaction, redox and oxidation, because these are happening simultaneously. So we have, so what was reduced? What was reduced to make hydrogen peroxide? You can think about that. It's because we know that something was oxidized, the deamination, long chain fatty acids, that was oxidized, so something had to be reduced. Something gained hydrogen, and it's oxygen, of course. Oxygen gains hydrogen to make hydrogen peroxide. And in the peroxisome, because hydrogen is going to oxygen, producing hydrogen peroxide, hydrogen peroxide is not a safe thing to keep. So the peroxisome is also capable of degrading hydrogen peroxide, producing and degrading it. And of course, the enzymes that are used, because sometimes enzymes come up, a little bit more rarely, but, of course, enzymes usually end in A's, and their names usually indicate their activity. So peroxidases. So we'll do the job to help in the transfer of the reduction and degradation of hydrogen peroxide. Okay, I think that's fine. We're going to move on to, I'll show you that. So that was question 6. Obviously, the answer was C. And if you happen to have the Biology Master Series book, background information in Bio 1.2.1. So now moving on to question number 7. Daughter cell by mitosis, and cytokinesis have. And then we look at various options in terms of chromosomes. Okay, so, the famous mitosis versus meiosis is really what they're trying to get at. And so what are these things for? Well, mitosis really is about replication. So it's your body cells, like somatic cells. Your body cells need to make more of themselves, either replacing, like in the skin, which is continually occurring, or if there's there's dead cells or something like that, or growth or whatever. So, but it's replacing, and that means it's creating two identical cells from one cell. So you're going from 1 to 2, and those two are identical. So normally these are diploid cells that are engaging in mitosis, producing to produce a pair of diploid cells, as opposed to meiosis. So meiosis, first of all, there's some things that should, just flow as soon as you are thinking about meiosis. And one thing about meiosis is the very famous reduction division. And sometimes they can ask you what part of meiosis has the reduction division, and then they can go through all the different stages, the first and the second part. But it is meiosis 1 is where we have the reduction division. And this means that we can start with something that is diploid and end up with haploid cells. So going from 1 diploid cell to 4 haploid cell at the end of meiosis. And of course, the reason for this is the production of gametes. You need to reduce the number of chromosomes to create gametes, ova, sperm, egg, sperm, in order so that when there is fertilization, that you reconstitute the normal number of chromosomes. So you need a process to have it so that when there is fertilization, the normal diploid state is reproduced. So mitosis, of course, this is, I don't know, grade 9, grade 10 high school question. Mitosis, of course, after cytokinesis, which is the development of the two new cells, produces the same number of chromosomes present in the parent cell. So then we have, for question 7, the answer is C. And if you have the book, you can go to Bio 1.3 and 14.2 for background information. Okay, now starch, cellulose, and glycogen are all what is starch, cellulose, and glycogen. So hopefully you recognize that these are carbohydrates, and that carbo stands for carbon. Big surprise. Hydrate is just like when you hydrate yourself, what are you doing? You're ingesting water. So carbohydrate molecules. And so their formula, you see the recurrence of carbon and H2O in their molecular formulas. And so they're definitely not proteins. And they are not linked internally between the molecules by hydrogen bonds. Of course, they engage in hydrogen bonds. No question about that. As long as you have an oxygen bonded to a hydrogen, which carbohydrate definitely has, then you'll be engaged in hydrogen bonds, but not within the molecule. And water solubility, actually, they do engage in hydrogen bonds, certainly around the surfaces of these molecules, but within these very complex extended networks of glucose polymers, they create, because it's a storage unit, imagine it that way, they create a molecule that is in its entirety not soluble. So it's a storage unit. And so, yes, answer choice D, polymers of glucose. some background information, 17.3, 20.3.3, but I think we'll just take a look at a couple of things. One, and obviously they're not nucleic acids like DNA and RNA, polymers of nucleic acids. A couple of things. One, question 4 of this same test also overlaps with this. And this happens sometimes on the exam, in which you'll get clusters of questions related to a general topic and then move on to a different cluster of questions. So you'll see that throughout this exam and throughout other exams from the ADA. And I just will look at, okay, let's take a quick look at 17.3. Yes, I just want you to put this in the back of your mind as well. You know, glucose is such an important carbohydrate, you know, both in photosynthesis and being the primary fuel for cellular respiration. But I also wanted you to, you know, remember about sucrose, because of course, sucrose is, can't say in human biology, you know, it's a great focus, but in plant biology, which you need to know for this exam, sucrose is famously the translocatable carbohydrate, which means that it moves through the phloem of the plant. So it's important to keep in mind. And of course, both starch and cellulose are structural carbohydrates and can be used in storage. I do want to show you one other table from organic chemistry. It gives a good summary of the story. So this is from Organic Chemistry 12.1.3. Okay, so first, the generic condition. I know most of you know this backwards, forwards, but just in case, monomers are the building blocks in order to produce dimers, 2, trimer, 3, tetramer, 4, or oligomers, which is some other number, some greater number or ambiguous number of monomers. And these are all combined with covalent bonds, not hydrogen bonds, in order to produce polymers. Now, to be more specific, amino acids, when they polymerize, they form, of course, dipeptides, tripeptides, tetrapeptides, oligopeptides, etc. And these bonds are called peptide bonds. In organic chemistry, they're actually normally called amide bonds, which are identical, but in biology, they're normally called peptide bonds. And so this, you can create your polypeptides or proteins like insulin or hemoglobin. And then for sugars, we should specify sugar. First of all, sugar is not a good word to use in science because it's more a colloquial expression. But if you wanted to talk about something that was a monosaccharide, you'd have to say simple sugar, because sucrose would also be called a sugar, but it is a disaccharide of glucose, fructose disaccharide. So simple sugars can combine, of course, to create disaccharides, trisaccharides, tetrasaccharides, oligosaccharides, etc. And these bonds are glycosidic bonds. So the covalent bonds for amino acids, peptide, saccharides or sugars, glycosidic bonds, and nucleotides, phosphodiester bonds. And you can see the same story there. Okay. I think that's that. So now we move on. So yeah, so we have polymers of glucose. I think that is pretty clear. Okay, so question number 9, each of the following cell organelles have a membranous structure. So we're thinking about the famous bilipid layer with phospholipids and the phospholipid heads and the tails within the membrane that are hydrophobic, and the heads that are hydrophilic, water-loving, facing either the extracellular space or the intracellular space, the cytoplasm. So now we're thinking about that same membrane around these different organelles. And so, of course, famously, answer choice A, the Golgi complex, famously has a membrane. It's a stack of membranes, and you can remember the Golgi complex, also sometimes called Golgi apparatus, depending on the version. The ADA has used both of those expressions, and you can remember it as being the export department of the cell. So, of course, the nucleus has a royalty in the nucleus. DNA tells the cell what to do. Messenger leaves, goes to the either to the cytosol with the free ribosomes to make proteins within the cell, or to the rough endoplasmic reticulum to make proteins that are usually associated, the rough ER associated with the Golgi, so that those proteins can go to the plasma membrane to be used as intramembrane proteins, or can be exocytosed and be used in the extracellular space or the intravascular space, like antibodies or something like that. So that's all the Golgi complex. Then we have centrioles, which we've already discussed, cylindrical, 9 triplets, in pairs, used in mitosis, meiosis, and not membrane bound. There is no plasma membrane around these microtubular containing centrioles. And then answer choice C, mitochondria, very famously double membrane. It's one of the 2 organelles within the cell that have a double membrane. It's the nucleus and the mitochondria have a double membrane. And be careful about any wording with how many membranes do you cross to get from, what is the, what is the minimum number of membranes to cross when going from outside of the cell to the inside of a mitochondria. Just have to be careful with the wording. If they ask plasma membranes, then okay, that's a bilipid layer, so it's 3. But they could find a way to ask how many membrane, phospholipid membrane layers, and then it's 3 times 2 or 6. So you have to be careful. You know, are they talking about the bilipid layer, or are they talking about each individual layer, which is a little, anyway, it's not a polite question for polite company. Then answer choice D, lysosomes. Lysosomes, I said before, you famously can remember them as a suicide sacs. They contain the content necessary to digest the any portion within the cell, which means that you're going to cut it up into their individual components so that you can reuse those components for the good of the cell. And finally, endoplasmic reticulum, which you can imagine is the synthesis center of the cell. And could be rough, could be smooth, but we talked about the rough endoplasmic reticulum being important for protein synthesis. Okay, and there's overlap between this question and question 6, and that does happen on the exam. You don't have to get nervous or freaked out about that at all. Okay, so that's question 9. The answer is B, and you can find that background information at those references. And finally, for today, question number 10 in anaerobic glycolysis. So we've set the stage. There's no oxygen available because it's anaerobic as opposed to aerobic. Doing some aerobics exercise takes up a lot of oxygen. Okay, but that's not the case here. So there's no oxygen. And this is, the truth is we actually answered this question fully last time when we looked at questions 2 and 3, which are directly related. Question 2 and 3 within this exam, which are directly related to this question. And we saw how glucose is divided, because think of the word glycolysis, glyco referring to glucose and lysis referring to breaking apart. So you break apart glucose. Glucose famously is a 6-carbon sugar, and you break it apart into 2 3-carbon sugars or trioses. And so let's just take a quick peek at that again. And we will see how our lactic acid, answer choice C, is being produced. And so I will just take a quick look at the summary in Bio 4.5. Okay, so this is just a quick summary. So this is what we're referring to in this question, anaerobic, without oxygen. So this is occurring within the cytosol of eukaryotic cells. And we have, of course, with oxygen, which will occur in mitochondria. And then we get more ATP per molecule of glucose for this 1 molecule of glucose. This is an exaggeration, theoretical level. It's much less than this. But what it does do is to underline that this aerobic pathway produces obviously far more energy per molecule, not necessarily 18 times, but far more energy per molecule of glucose in reality. And it also famously produces the waste products of carbon dioxide and water. And that is what you see in organic chemistry as the results of complete combustion of hydrocarbons. So this is quite normal. So, of course, this is a longer pathway, takes more time, but far more efficient in terms of the result. Now, anaerobic, glucose, glycolysis, meaning to break apart into 2 3-carbon compounds, pyruvate, and 2 ATP. Now, this is the part that's called fermentation. Of course, if it's in a plant, that fermentation is going to result in alcohol. Famously throughout human history, some rotting foods, somebody discovered that, hey, this you can drink it at a certain point and not get too sick. That's the human discovery of alcohol. So of course that's through plant fermentation, but through animal fermentation, we get the very famous pyruvate. Pyruvate through glycolysis. Also described as pyruvic acid, fermenting to create lactate or lactic acid. And this, of course, occurs relatively fast compared to anaerobic respiration. And so we get 1 mole of glucose producing 2 moles of lactic acid, which is answer choice C. The carbon dioxide only comes as a result of the aerobic respiration in the presence of oxygen, and that's answer choice A. So that would be aerobic respiration through the electron transport chain, Krebs cycle, etc. And then answer choice B, 2 moles of acetic acid. Yeah, that's just a different thing entirely. Acetic acid is not an important part of this process, though acetic acid is known to inactivate glycolysis in the liver. But that's a very specific thing, and that's not it's not what glucose is oxidized to. And so answer choice C, then answer choice D, of course, so acetyl coenzyme A. So that's the important link here, where the 3-carbon compound is able to send 2 carbons into the mitochondrion, where we have the Krebs cycle and then electron transport chain, and that link is acetyl coenzyme A. And yeah, answer choice C is crazy and removed from the new exam. So because the 2022 exam, for the most part, removed answer choice E for the natural sciences. DAT, PAT has remained the same in terms of which subsections have 4 choices and which subsections have 5. Okay, well, that's it for today. You know, I hope you learned a little bit, and I hope that most importantly that you start getting into a habit of regularly preparing for the exam, or regularly studying for the exam, using different resources, and yeah, and if you do that over a couple of months, you can get the score that you're looking for with your efficient studying. Okay, so that's it for now. We'll see you again next week, and we will go through another 5 questions. And if it does go relatively fast, then we'll start doing 10 at a time. So we'll see how that goes. Good luck with your studies and stay safe. Bye for now.